This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 474, recorded on December 29th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today in New York City, where it's been very cold, minus 10 Celsius today, Dickson Despommier.
Hello, Vincent.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it is brutally cold.
Yep.
What do you got?
It's currently gotten up to minus 9 Celsius, 16 Fahrenheit.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's a little warmer today than it has been. It's 18 Fahrenheit, which is minus 8 Celsius.
All right, this is the last TWiV of 2017. How about that?
It is.
We're going to do the next episode in a week from today will be our summary of 2017. It'll be 5 minutes long. I don't think so.
No.
But today we will do it. We'll do something slightly different. But first, Kathy, any more word from ASV?
Yes. The registration part is live, and before you go and actually register, you might want to think a lot about whether you want to attend one of the satellites that are sort of the pre-meeting events on Saturday morning and early afternoon. There are either 5 or 6 of them, and you can check the webpage out and see which one you might want to attend and sign up for that one. And then there's kind of this unwritten policy that once you're signed up for one, if there's a particular talk in another one that you want to go to, you can do that. So that would be something worth thinking about. And then, of course, the usual reminder to get your membership up to date so that you can submit your abstract, sponsor an abstract, or you don't have to be a member to submit your abstract, but if you want to apply for a travel award, then you do. And if you want the reduced registration rates, you should be a member.
Meant to say that we are going to give away a book today, and we have a different kind of a contest. Mm-hmm. This will require some work on your part. And immediately everyone's shutting off the podcast.
Right.
Oh, well.
This is a nice...
I don't need your stinking book.
This is a nice cartoon. Who posted this here?
I did. I just saw it, and I love it, that it lists all the horrible things that happened this year, and it's Fraz, and she says, name one good thing about this year, and Fraz says, it was a prime number, and so that means that only mathematically it won't repeat itself for another 2017 years.
Yeah. We cling to whatever floats.
Ooh, bad idea.
Really?
Bad idea.
All right, we have a few follow-ups. First, Anthony sends us a reminder that on the 27th of December, which was 2 days ago, in 1831, the HMS Beagle set sail.
Cool. And
there is a Royal Society Facebook page which talks about this. Who knew the Royal Society had a Facebook page?
Hey, they're modern.
And
Sure.
of course, you can tell that they are at Carlton House Terrace in London. You can see exactly where they are. That's probably a nice location, isn't it?
Yep.
And, so that's
I had lunch
pretty cool.
at the Royal Society. It's a wonderful building. In fact, we ate under the portrait of Charles Darwin.
Really?
Yeah.
Was it inspiring?
It was wonderful. Yeah.
Did you have tortoise soup?
We had something like finch salad, I think I was.
Okay.
Fish salad. All right. Now, Anthony also has a follow-up. Concerning the comments on TWiV 473, I already knew that confirming that a case of dengue was a first infection could be done. I also knew it should be done if one wanted to gauge the severity of an initial infection. At least that's how I read it. Whether that indeed is being done or if self-reporting is being relied on, that I don't know. I did an admittedly all too quick skim of Google results. I hoped that there would be clear statements of the steps taken to confirm that severe first-time infections really were that. This I did not see. I've actually been thinking of this for a while. Perhaps you might remember my neighbor whose family became sick while on a trip home to India. I asked him if it was the flu, and he replied, no, it was viral. That's great. The topic of dengue came up in the conversation. I mentioned how the first time dengue was relatively mild. He then said, that's not true. My sister only had dengue once, and she never was so sick in her life. I didn't argue with him, but I did wonder to myself how his sister could be sure that this indeed was an initial infection.
Right.
For everybody, a passing slight digestive upset, a headache, muscle ache, or perhaps just some out-of-sorts feeling is common. This generally is just thought of as some effect of an unpleasant commute, boring meeting, long day at work, something eaten, etc. How often is this actually an invasion by a pathogen that the immune system manages to suppress quickly? Well, it could be, yeah. You could have little innate immune responses that clear pathogens, and maybe you just feel a little out of sorts because of that interferon that's coursing through. Anyway, so now I did a proper investigation, and I first went to the dengue chapter in Fields Virology, which writes, epidemiologic data in Thailand has shown greater than 10-fold higher rates of dengue hemorrhagic fever, dengue shock syndrome during secondary compared with primary infection in children. So I went to the reference, and this is a paper published in 2000. In this paper, they studied children with acute dengue in Thailand. Patients with a secondary antibody response were twice as likely to have dengue hemorrhagic fever compared with those with a primary antibody response, 53% versus 23%. Okay, so there they did a very careful study. And yes, you can have some primary infections have dengue hemorrhagic fever. Dengue shock syndrome was associated with a secondary dengue virus infection in all cases. So in this small study, they didn't see dengue shock syndrome in any primary dengue infections. What the main point of this paper, though, was that they showed that high viremia titers 3 days after the onset of fever correlates with severe disease. And that would make sense if you consider that the antibodies are allowing the virus to enter new cells and get higher titers and so forth. By the way, also interesting, babies who get primary dengue can also have severe dengue because they have maternal antibodies from a mother's previous dengue. So it's almost like having a secondary infection, even though the babies were only infected once. So that's a careful epidemiologic study. And no, they don't do that every day. If someone goes in the hospital with severe dengue, they take care of them. They diagnose it as dengue and they take care of it. They're not going to say, is this your first or second infection? Only in certain studies, which cost money and take a lot of time, will they do that. Does that answer the question, you think?
I think so.
I think so.
But I think Anthony wants to know in every case of severe, is it a primary or secondary? We're not going to have that information. Nobody's going to do that. You have to do some science. That's the epidemiology, and that doesn't get done all the time.
Because you have to do the medicine.
Right. All right. So I thought for this last paper of 2017, actually, I did look at a flavivirus paper, and I said, no, I can't do another one. Because I promised no more dengue, and so close. But here we have a very interesting paper, which is in bioRxiv. And I do this for two reasons. First, I thought we needed a DNA virus because we do lots and lots of RNA viruses here. And this is a giant virus. We like giant viruses. And finally, just to show that you put something in bioRxiv, we can talk about it. It doesn't have to be peer-reviewed. And I like the bioRxiv idea. And so I embrace it. I think everyone likes bioRxiv here on this show, right?
We are a form of peer review, right?
For sure.
Okay.
And we can do that.
And this paper,
we will do that.
This paper does have issues. And I know Kathy will note lots of typos, right?
That and some other confusing parts. I'm thinking ahead of time, if they haven't submitted it and the authors want to contact me for what I thought was confusing, I'm happy to provide that information to them.
Great.
So, and because it's bioRxiv, that means it's open access. Is that correct?
Yes.
Everybody can see it. Creative Commons version 4. You could even use figures from it in your own blogs or podcasts if you wanted to, as long as you cite the original authors. And I think that's just great. All right, so the paper is called the kinetoplastid infecting Bodo saltans virus, BSV, a window into the most abundant giant viruses in the sea, by Christoph Dieg, Cheryl Emilian Chow, and Curtis Suttle from the University of British Columbia and the Canadian Institute for Advanced Research. Now, we love giant viruses.
Yes.
Here, we're fascinated by them. And this is another paper about a giant virus. Curtis Suttle has worked in this area for a long time. He was on a TWiV a number of years ago. You can find that where he talks about going out on big ships and collecting samples of water deep in the ocean. And so he's the PI on this paper, which finds a new virus that infects. Here's the executive summary. Bodo saltans is the name of the organism, and it is a kinetoplastid. And we'll talk more about kinetoplastids and what Bodo saltans is. But it is in the family Mimiviridae, but they think it's a new, what is the taxonomic level that they're putting it? Aquavirinae, a subfamily within the Mimiviridae. All right? Is that a good summary to start? And it has some interesting features that make it different from all the other giant viruses. Yeah.
Right. And they're saying it's the first closnovirus isolate.
That's right.
And these were proposed, or there's in silico evidence for them, or metagenomic evidence for them. And we talked about that on TWiV 437, I believe, is the one.
Yeah. That was Kathy's new spindle virus.
Right, where they identified closnoviruses from metagenomic data. And so this is the first actual virus.
Yeah, so in fact, after that paper, which included Eugene Koonin as an author, the Jean-Michel Claverie and Chantal Abergel wrote and said, they actually don't have virus, they just have sequence. So who knows? now this we now have a Klausno virus. That Klausno sequence was from a Schmutzdecke, if I remember, something like that, at the bottom of a sewage treatment sludge pile.
It was some kind of
nasty place where
starting sample,
primordial ooze. So this so what they did here, part of the issue here, well, first let me just say this amazed Dickson. First of all, they start the paper by saying viruses are the most abundant biological entities on the planet, and I love it because they didn't say life forms, which some people do. So Curtis Suttle has got the right Kool-Aid. And then there are typically millions of virus particles in each ml of marine or fresh waters that kill about 20% of the living material by weight every day. Incredible.
Is that a lot? How many pounds is that, Dickson?
Gigatons, it's more than that even, I'm sure.
So that means they're releasing a lot of material.
Incredible.
And, that can go on and be cycled, right?
80% left every day. That means that the division cycles are pretty fast too, to replenish all of that.
I think the number is 10 to the 30th infections per second in the ocean. Per second. As we're sitting here, just
mind-blowing.
Yeah, it's incredible. It's a lot. So most of the giant viruses, so the oceans are full of viruses, but there are also lots of big ones there, and most of them have been found to infect Acanthamoeba species.
Right.
With three exceptions.
So just to be clear, this 20% of the living material, it's not like 20% of the fish that are being killed. This is single-celled organisms are getting killed for the most part.
That's right.
It would be funny if fish were being lysed at that rate.
No.
Wouldn't be funny at all. No.
Oh, but they die too.
Humans are working towards something like that.
They die too, by the way. The fish die, of course they do. Everything dies and then they sink to the bottom, right? And so the oceanographers have a wonderful term for this, because there's a bunch of organisms that live below the photic zone that have no option for grazing or, filter feeding and stuff like this. They call it a rain of carcasses.
Yes.
And they feed on dead things that come down to their level and they eat them. So things get consumed and then consumed and then consumed and then consumed.
It's a food web, isn't
it? And then subtended underneath the plates, they're migrating around.
The circle of life.
So keep singing it.
These giant viruses, there are only 3 that have been found that do not seem to infect amoeba. That's Cafeteria rowenbergensis virus, that
affects the
Infects
eponymous
salamis.
and the phytoplankton infecting viruses, Phaeocystis globosa virus, and Oreococcus anophageferens virus,
Which infects cookies.
Phytoplankton infecting virus. What's a phytoplankton? A plant, right? A small plant.
A very small plant, like
a
unicellular.
a small floating plant.
That's
A
right.
small
That's
floating plant, as opposed to a zooplankton, which is a small floating animal.
So the zooplankton feed on the phytoplankton.
Is that right?
That's right.
And on them, they're smaller and smaller.
No. That's well. Then you got the viruses, of course.
Then the viruses infect and kill 20% of them.
Exactly.
Right.
So the authors state that they were motivated to find ecologically relevant giant virus isolates that infect microzooplankton. So they say in the introduction, we isolated and screened representative microzooplankton in order to isolate new giant viruses. Okay, and that's where Bodo saltans virus comes from.
Right.
Bodo saltans is a flagellated eukaryote in the phylum Euglenozoa. Euglenozoa. Is that good? I don't know how to say it.
Euglenozoa?
Euglenozoa.
That's how I would say
That's
it.
better because euglena, everyone knows, right?
That's right. Yeah, well, not everyone.
You glean a little here and there.
Ooh.
And so in freshwater, the bodonids are part of this, like Bodo saltans. And then in the oceans, they're diplonemids, which Dickson thinks means they have two flagella.
They do.
Yes, one in front, one back.
Now, I happen to have in front of me
No, they're both coming out of the front.
Is this a euglenozoa, this thing I'm holding, this leishmania?
It's a promastigote, so it's reminiscent of the euglenas, but it's
So wait. So Wikipedia has Bodo saltans wrong. It says it has two flagella, a short anterior one and a longer posterior one.
Well, something else said that it has two flagella, one oriented posteriorly and one oriented anteriorly. So
they
they
all
come out of the same
place. They come out of the same place.
and point different ways.
That's correct.
Okay.
That is correct.
Anyway, I have a giant microbe leishmania.
You do.
And it has a flagella, which
that's
is this
the form
cord,
in the sandfly that you're holding.
and they put the eyes at the wrong end of the leishmania. Anyway, these are kinetoplastids, these organisms, which, because they have a kinetoplastid, which is a mitochondrion with circular concatenated DNA, which Dickson and I spent an hour discussing heatedly, right?
Well, I already knew, but you wouldn't believe me.
What is a circular concatenated DNA?
Well, there are two kinds of circles inside the kinetoplast. Okay, two kinds. There's a maxi circle, and there are about 10 to 15 copies of that, and those encode the proteins that are mitochondrial in origin.
Like the respiratory chain.
But it's not a complete story. It's not as complete as our mitochondria, for instance, but it supplies a electron transfer chain. Then there are the mini circles, and there are thousands of those. And those are the concatenated.
Are they linked?
They're linked like chainmail.
Chainmail. Okay.
I was just going to say linked like chainmail.
Well, that's what we read, but Vincent wouldn't believe me. He says they can't be linked together.
Kathy,
but they are.
When you have rolling circle replication, you have concatenated genomes, right?
Yeah, but
and that's what I'm thinking about. They're not linked, but
no, I think the word refers to a repeated something.
Yeah.
So when I was at UCLA for my postdoc, we had floor meetings, and Dan Ray was on our floor, and he worked on Crithidia fasciculatum.
There you go.
Which is a model organism for studying these kinetoplast DNA. So we heard about maxi circles and mini circles a lot.
Did you know Larry Simpson?
I knew of him, but I mostly knew Dan and the people in Dan's lab because they were right across the hall.
And
do the mini circles become linked as a result of replicating, and then the copy is linked to the one that was there previously because the replication produced that?
Apparently. The replication of these things is
so they're not resolved by they're not resolved by topoisomerases, basically. So when SV40 replicates, they're separated in the end by topo.
And these are not separated.
But there are topoisomerases that are quite important and are speculated to be important targets for the kinetoplastids, the organisms that are pathogenic.
But obviously they're not resolving the concatemers at the end.
They're doing something that's causing this linking and concatemerization.
So
now I'm thinking at some point that this you must be able to separate these in the cell in order to produce another kinetoplastid, right?
Go talk to Ken Stewart.
So when you take a circular DNA and you replicate it from say a bidirectional origin, in the end you're going to have two concatenated circles. In eukaryote, in cells infected with SV40, say, topoisomerases cut and reseal the DNA so that the two circles are separated.
Right.
So there may be topos in these kinetoplastids, but they're not.
There are, and they are doing things. It's complicated,
but they're not separating the two circles. That's
no,
what
they're not.
They might be untwisting them and putting them back together in this chainmail kind of thing.
Okay.
So each one,
and there's some mechanism that I gather don't quite know that allows this whole system to produce an offspring, right?
So I found an interesting figure in eukaryotic cell. I linked to it. Kinetoplast DNA network, an evolution of an improbable structure. It's a picture showing proposed stages in the evolution of a kinetoplast DNA network. A, supercoiled, non-concatenated minicircles. B, present in early branching bodonids. B, relaxed, non-concatenated minicircles, also found in early branching bodonids. C, small catenanes of relaxed minicircles, relatively abundant in some late branching bodonids. And D, the kinetoplast network present only in late emerging trypanosomatids. So if you look at the phylogenetic tree of bodonids, you can see different stages of this evolution of the concatenates.
Yeah, so these minicircles encode guide RNAs, which leads to the expression of the proteins from the maxicircles. Mm-hmm. Okay. There's a lot of RNA editing that goes on. There's a lot of stuff. I spent 3 months in Ken Stewart's lab once, and that's all I heard too, Kathy, is
minicircle, maxi.
Mini, maxis, minis. And
all right, so this it doesn't come back to this discussion, but it would be interesting to
exactly.
Right. And my first comment on this manuscript is it would have been nice had they just mentioned that the kinetoplast is this different kind of mitochondrion that's right. Found in this organism.
Exactly right.
Just for the people who are not cognoscenti and haven't heard about it forever. Right.
Now, I want to talk a little bit about the isolation because I think it's not clear to me. Maybe all of you can sort it out. They say to isolate giant viruses, we isolated protistan microzooplankton from a variety of habitats and screened them against their associated virus assemblages. Now, what they actually did is they collected fresh water from ponds in British Columbia, and I think they had 11 freshwater locations in southern British Columbia. They had 20-liter water samples. They concentrated them. And then they used that to infect the host organism, which was isolated near the University of British Columbia, near campus, Bodo saltans, okay? So why did you take 20 liters of samples from other places and then got Bodo? Did you know that where you got these water samples from, there was Bodo saltans there, or because that's not mentioned at all? It turns out that the virus does infect Bodo saltans, but the virus came from a different place. So I don't understand the statement.
They may have looked close to them first and didn't find it, although they found the host. And then they started to look around for the virus.
So that's what this statement screened against their associated virus assemblages. I don't understand that because the virus and the host were from different places. Anyway, they take these concentrated samples, they put them on Bodo saltans, and it replicates. Right. They get quite a nice titer viral concentration, 2 times 10 to the 7th particles per mil, and they monitor the host cell density by flow cytometry. Of course, you can't do a plaque assay on these Bodos because they move around. Got to make a monolayer, right?
You could put them in a kind of a gel matrix like you do for some cells. Yeah, sure, you could probably do it.
And you could hold them in place.
Yeah, you might.
Anyway, so they have a virus that infects Bodo saltans, and it's a big virus. You look at it in the electron microscope, it's 300 nanometers.
It's huge.
It's very big. And it looks like a mimivirus. It has it's an icosahedral capsid, and it has many layers. It has a halo surrounding it. So in the pre-show, we were talking about needing more angels. Well, here we have a halo at least.
That's right.
It has a Stargate capsid-like structure, which was found
Go right to heaven.
in mimes that may be involved in releasing the DNA from the capsid. So it looked
and
like
sending it into another dimension.
Exactly.
Sending it to another dimension. And so they do some imaging of infected cells. So they take Bodo saltans and infect them and take electron micrographs, and you can see particles assembling in these Bodo saltans.
Not too many, though. I mean, you can count them with one hand.
It's true. Not a lot. They say they replicate in factories. And this is interesting. The factories were always observed in the cell's posterior. So it would be opposite to the flagella. Is that correct? Yes?
That's correct.
Wonder why that would be. You think things want to stay away from the flagella?
No, I think maybe there's some order to the cytoplasm of Bodo that this virus takes advantage of.
Okay. As infection progressed, the Golgi disappeared.
Aha.
The nucleus degraded.
Aha.
But the kinetoplast remained intact.
Right.
And the factories, this term virus factories, this is where the DNA is replicating and the virus particles are being assembled, first observed at 6 hours post-infection. And 18 hours, the first mature particles observed. So it's not too slow, right?
Nope.
Not bad. Nope. All right, so that it looks like a mimi virus in all these features. They sequenced the genome, of course. 1,385,869 base pair, linear, double-stranded DNA.
Woof.
Exactly.
It's a lot of bases.
Come on, we're going to talk about this because
Yeah.
Now this is if you go to giantvirus.org, which has got to be one of the most neat virus websites you could go to. The top 100 known viral genomes, and this is now going to be number 3 because the two biggest ones are Pandoravirus salinus at 2.4 million base pairs, Pandoravirus dulcis at 1.9, and now we have this one, number 3. So it jumps to number 3 on the list of giant viruses. And so just for that, it probably will get into a luxury journal, I guess. Who knows? 1,277 predicted open reading frames. So those could be proteins. 27% of those match protein sequences, known protein sequences from eukaryotes. And these include sequences in the host, Bodo saltans, from bacteria or archaea or other viruses in this group, by the way, which is called NCLDV group of viruses, nucleocytoplasmic large DNA-containing viruses.
An abbreviation they did not spell out in the manuscript.
They didn't spell out a lot of abbreviations, didn't they?
But they will.
Yeah.
See, they're actually vying for free editorial services.
So the essential genes include those for replication, translation, DNA replication, the structural proteins. They're all in the central part of the genome. And at the ends, which they call the periphery of the genome, you got duplicated genes, including 148 copies of ankyrin repeat-containing proteins. We'll get back to that, because I want to tell you that 54% of the open reading frames, they had no idea what they did. They didn't look like anything on Earth, and that's as far as we can go.
On Earth. Now you've touched on an interesting subject.
And we don't have sequence for anything not from Earth.
Yeah.
Right.
You think we will?
That we know of.
You think we will? If someone brings back sequence from Mars, someone will say it's a contaminant from Earth.
Yeah, they will probably.
Dickson, is there water on Mars?
Yes. Of course there is.
So they have lots of genes for DNA replication and repair, transcription, the synthesis of mRNA, and the processing of mRNA capping and poly-A addition and so forth. They have genes that might be involved in membrane trafficking. Of course, they have 4 major capsid proteins. They have metabolic proteins. This really excited Dickson today.
It sure did.
Likely involved in carbohydrate metabolism. And Dickson said to me right away, this can't be a virus.
No, it can't.
We got in a big argument.
I'm going to say it again. It can't be. This is not even an atypical virus. This is a degenerate rickettsia.
All these other giant viruses have lots of enzymes.
You know what? Then maybe this is wrong to call them giant viruses.
Well, Dickson, that is one of the 2 theories for where these viruses came from.
Why don't you call them mini What was that other word I was looking for? Rickettsia. Mini rickettsia rather than maxi viruses.
Well, even a rickettsia can do some things on its own.
Of course.
This virus can't do anything.
You can't culture rickettsia. You can't culture rickettsia outside of a cell.
Rickettsia do encode ribosomes, though.
They're an advanced virus.
No, they're not. It's not even close.
No, I'm not saying they're close, but they're reminiscent. They have metabolic genes. There's no need for that on a
and this is one of viruses. Right. So this is the two camps in the giant virus field are the people who say that these are degenerate organisms that degenerated into viruses, and the folks who say no, they're additive viruses that acquired and just kept acquiring, and they grew to this size.
Dickson, I feel that you have not been present for all the discussions.
No, I was present for all of the discussions on the maxi
maxi circles.
No, not the maxi circles.
The maxi viruses.
Stop it.
The two giant virus TWiVs that we did, 437 and 440. At least one of those was putting one of the nails in the coffin of the alternative hypothesis. And this one, I think, also does too, shutting down Dickson's argument.
Oh, yes. You're shut down.
We'll get to that.
Shut down. This genome also encodes.
That's what I was kind of setting up there.
I'm outnumbered 3-1 here. What do you expect?
You're not outnumbered. No, it's not about numbers. It's about how well you think.
I'm thinking out of the box.
Now, this device also encodes a complex translation machinery with lots of initiation proteins, as do the other giant viruses. However, it doesn't encode any tRNAs, and all the other giant viruses do. Now, when that was first discovered in a giant virus, tRNAs, initiation proteins, tRNA synthetases, people were stunned because none of that was ever found in a viral genome. But of course, it doesn't have a complete translation system. You cannot translate, but you still depend on the host cell in order to do that. Now, why aren't there tRNA genes encoded in this Bodo saltans virus?
Why aren't there?
I can't ask that question.
I don't think so.
What's the reason? Could I say what's the reason?
What is the selective pressure not to have?
You actually must have listened at one point. What is the purpose of not having tRNAs when the other viruses do? And they speculate that is because these kinetoplastidae undergo extensive RNA editing.
They do.
Can you tell us what that is, Dickson?
Well, like in general, but I'm forgetting some of the details. So why don't you fill us in on the details?
Well, it's a
uridine to cytosine or cytosine to uridine?
No, it's just adding bases that are not templated.
Exactly.
In the DNA.
Yeah.
Okay, by whoever mechanism, slippage and so forth.
Yeah.
And these kinetoplastidae do it. And that Ken Stewart, is that his name, Ken Stewart?
Ken Stewart.
In Seattle. He's been studying this extensively with trypanosomes, right?
Yes, that's correct.
So they speculate that because of that, it cannot encode its own tRNAs because the virus cannot carry out RNA editing. So the tRNAs in the host are extensively edited, and therefore this virus, which infects this host, can't carry their genes. But they do have repair genes for tRNAs.
Yeah, I wanted to know more about tRNA repair genes, and they didn't give a reference. And then they list a bunch of them, and so you can kind of glean from that the kinds of things they do. But evidently it's a whole category or genre that I was just unaware of. So an RNA splicing ligase, CAA nucleotide transferase, phosphotransferase, hydrolase, methyltransferase, pseudouridine synthase, etc. And says they appear to have been recently acquired from the host, and they don't really relate that to the tRNA, the absence of the tRNA.
Yeah. Recently acquired, meaning that there are very few changes in the sequence compared to the host.
That's how you would say that.
Right.
Right. Yeah. Now, these duplicated genes, 148 copies of ankyrin repeat proteins. Now, ankyrin repeat proteins may be involved. They speculate in the discussion later. They may be involved in antagonizing a host immune response against the virus. Now, they call this genomic accordion. Now, and we talked about that on TWiV a while ago. This was first discovered by Nels Elde and Harmeet Malik's lab. That was, what TWiV was that? I wrote it down, but I can't see it. So in vaccinia virus genome, there is a gene that antagonizes the host defense protein, PKR, protein kinase RNA activated.
Pox has got a squeeze box.
There you go. Pox has got a squeeze box. And in under certain conditions, the vaccinia virus can amplify this gene, this antagonistic gene, get many, many copies, enabling the virus to replicate in the presence of the antagonist in the host cell. And then at some point, the gene undergoes a mutation, which makes it a potent antagonist. And now the accordion collapses. You don't need all those copies anymore.
See, I asked Vincent, why would it collapse when there's no pressure to collapse it?
Well, there may be pressure.
What's the pressure?
Well, I think one of the speculations in the paper, which I don't think you were present for.
A metabolic price tag to produce all those copies?
Extra DNA in your gene. You have to copy it, right? That costs energy.
Right. How much of our genome is not ours?
A lot.
Excuse me?
A lot.
Why haven't we gotten rid of it?
We need it.
For what?
Well, to make a placenta for one.
Yeah. There was a pause here, folks. I want you to know this is edited out, but that's going to be a pause.
I'll keep it pausing. There are other things as well. And in fact, we've talked about it. One of the cooler things was a paper from Nels Eldy's lab where they showed that a lot of the RNA transcriptional control elements for interferon genes are from endogenous proviruses. They're viral, and that's a good percentage of the genome as well. So,
by the way, that TWiV with the genomic accordion was TWiV 198.
Ooh, a long time ago.
September 2nd, 2012.
So, Dickson, the fact is that in Nels's hands, these amplifications come and go. You can see them coming and going. So there's obviously, it's not a good thing to have lots of copies of a gene in the genome unless you really need them. But you're right. We have lots of ancient, supposedly dead retroviral genes, and they're still there.
It raises another question. And if you're right that they have purposes, but you just haven't found out what they are yet.
We may not have found them, and people are looking and working on it. Right?
Can there be that many different things that we don't know what they do and still have that much of our genome?
You're asking, is there junk DNA?
Right.
And,
right.
I am.
Maybe.
That is another controversy. But the other thing is the constraints on replication for a virus are, I suspect, a lot more severe than the constraints on replication for something as huge and complex as a human.
Okay.
We have so much other overhead going on that a few extra genes, I mean, the amount of genome we're already copying, you throw in some extra, who's going to notice? Whereas if you are replicating a viral genome and you throw in a little bit extra, that's going to be a big deal. Replication cycle after replication cycle. That's I'm just
I have a
theorizing.
Didn't he just diss viruses?
Yeah,
he did.
I don't think I'm dissing viruses. They operate.
You said they have less overhead, but I don't know. Maybe they would have.
No, they're operating under tighter constraints, is what I'm saying.
True, but I've always wondered. When you look at plant genomes, for instance, they're huge and they're triploidy. A lot of them, and orchids have an enormous genome.
Yeah.
And they haven't gotten rid of the extra copies. They've got 2 extra copies of every gene in their whole genome.
We have yet to figure it out.
I don't get that.
I don't think we do either.
If I was Rich Condit, he says, I don't get that.
But the observation is these amplified genes in vaccinia virus, they get amplified and then they go away. It's clear.
No,
There's
protozoan parasites behave the same way. Leishmania has a lot of that.
And when a bacterium enters a mutualistic relationship with a host cell, becomes an endosymbiont, it loses genes that it doesn't need anymore.
However temporary the association.
Yeah, if it's not temporary, of course, they won't lose the genes. But if it becomes established, they will lose genes, and then they can't leave anymore. They're there.
Like mitochondria, for instance, or chloroplasts, for instance.
You bet. The other thing that's interesting about this viral genome is what is called the mobilome. And this is not a gas station. Is there still
Or is
mobile?
it a trailer?
It's not a mobilome. That's right.
Good. Excellent. An English trailer. A mobilome.
Mobilome. These are DNA sequences that move around the genome, right? Mobile elements. They can move between genomes.
Transposons.
Transposons would be an example. In other giant viruses, there are virophages that seem to move genes from one giant virus to another. And there's a transposable element called a transpoviron. Okay, it's a lovely name. But they're not present in the BSV. They have instead other kinds of mobilomes, which consist of inteins, autocatalytic proteinases, and self-splicing group 1 introns.
That's cool.
You know, I wrote a blog post about this late last night, and I didn't feel like explaining everything, right? So I just wrote these things out. And this morning I got an email from a guy who goes, usually I can understand this stuff. I don't know what you're talking about. Yeah, I'm sorry. I just was late and I had to go to bed.
Hey, dude, what you been smoking?
I said, I'll fix it. I'll go back and fix it.
So inteins are, another term for them is protein introns. They are protein sequences that splice themselves out of proteins and then reconnect the two halves of the protein.
Yep, it's cool.
What happens to the little piece missing?
Like an intron, right?
Maybe it does something.
Chewed up and recycled.
Most likely it's degraded, yeah.
Right.
Recycled, though.
I know you're not finished with this paper yet, so I'll save this for later.
Wow. Self-restraint.
I'm trying to restrain myself here because...
Go on, Kathy. Explain the other things.
Well, autocatalytic proteinases, I think inteins would be a subset of that, but an autocatalytic proteinase is a proteinase that works on itself. And so, but maybe it cleaves one end or the other and not necessarily the middle out and having to restitch it.
So it cuts a piece of itself.
Right.
Right.
And then self-splicing group 1 introns are a type of intron that can splice themselves out of an RNA. So we're talking about these things acting both at the protein level and at the RNA level.
So they find in the genome of this virus evidence for these elements, right?
Mm-hmm.
Now, one thing I didn't understand, maybe you can explain. They find them in genes like a DNA polymerase and I think RNA polymerase. They find introns in there and inteins. So have they gone in and then changed to maintain the open reading frame, or has it disrupted its own genes using this mobilome? Did you understand that?
No, this whole section precedes about 12 or 15 lines of text where I wrote, in the margin.
Okay. Because the thing is, like these genes, polymerase gene and DNA and RNA polymerase genes, they see evidence of these insertions. So I'm wondering if it's a duplicated copy that got inactivated or if it's still active. I can't tell.
I'm convinced that if Lewis Carroll was alive today, he would be a virologist.
Why is that?
Because everything is curiouser and curious. He's got all these crazy names for stuff that really are quite fanciful and poetic almost.
Yeah. Anyway, it's interesting. So there's a lot of gene movement, and this is another mechanism for that. They also say that maybe this mobilome helps to defend against, say, another virus infecting the same cell that it's in. Maybe it could send over its mobilome and inactivate it or something like that.
Right. That diagram is kind of cool that describes that with the red and green virus genomes. Or yellow and green genomes. Yeah.
I saw Dickson looking at it this morning. I was impressed.
Oh, yeah. It was quite very...
All right. So this is now, by phylogenetic analysis of this and all the other giant viruses, they're going to place it in a new subfamily of the Mimiviridae. So the Mimiviridae would be a family-level classification where all these other giant viruses, many of them are. And so this would be, what is it going to be called? Aquavirinae? Aquavirinae, because they are...
From water.
From water. And they say they have looked at the data sets produced from the ocean sequencing project, right? And they find lots of viruses related to this, so they think this is a really abundant virus, more so than other giant viruses, and maybe the largest group of identifiable giant viruses in the oceans.
Goodness.
And that's it. Together with Closnu viruses and Cafeteria rhombergensis as a distant relative, it defines a new clade of abundant viruses.
Okay, so here's where I want to interject. In the abstract, they talk about this being the first closnovirus isolate. And then there's one other place where they talk about this, BSV being a closnovirus.
Right.
And then toward the end of the paper, they start talking about it as if it's different from the closnoviruses. That really confused me.
Yeah, because if you look at the tree, it's kind of close to Closnu, but it's not a Closnu. It's separate from a Closnu.
But in the abstract, they say it is the first Closnu. So they need to clarify that.
It's a pseudo-Closnu.
I think they could say it's very similar to Closnu viruses, which would...
Or each time in the later part, if they just put other closnoviruses,
that's right.
This compared to other closnoviruses,
right.
Because they say they don't know the natural host of the other cosmoviruses. I inserted the other.
Yeah.
They just, so it's,
So the other thing here is that this virus was isolated from fresh water, right?
Right.
And they find related sequences in oceans, right? So, and that's why they say this could be like one of the most abundant giant viruses in all the waters. But
it could be.
They haven't actually isolated it from oceans. And is there Bodo saltans in oceans?
According to Wikipedia, there is.
I think it's found in both fresh and salt water.
Exactly.
All right, so it'll be interesting to try and.
Yeah.
Huh.
Exactly.
Now that may be that this species name has been assigned to two things that we just don't realize are separate species yet.
Mm-hmm.
Or it may actually be that tolerant of swings in salinity. I don't know.
Yep. So what I'm imagining here, just lay back on your bed and stare at the ceiling, is you think about the ocean as, when you look in certain parts of the ocean at least, it's clear as a bell. The tropics, okay? So it's a sterile environment basically there. I'm not kidding. It's almost sterile. If you go down to the poles, of course, then it becomes like a soup, because that's where most of the algal blooms occur. And I imagine the ocean is a gigantic soup of cell-free extract. Every 20% of all life forms that are microscopic are breaking open within a 24-hour period every day. That's an incredible enriched medium, don't you think?
Mm-hmm.
Sure.
Mm-hmm. And I would love to see some evidence for that from, let's say, a satellite that could sense something in the ocean that would tell you how soupy it is. Because, they can see the algal blooms and they can see the zooplankton blooms that follow that. But to see the result of all of that, to see the soup nature of the ocean would be a wonderful new addition to our ecological understanding of really the carbon cycle, because that's really what you're looking at here is the carbon cycle.
You mean when you say the soup nature, you don't mean the general turbidity. You mean
I mean the soluble portion that's released from cells breaking open.
You mean the amount that's right, that's from cells, but that is not in cells.
Exactly correct.
Okay.
I would liken that to a cell-free extract, basically.
Yes.
But it's missing the portions that were put into viral
How much of the ocean is cytoplasm?
Exactly. That's, Alan, perfect way to put it.
Yeah, well, the lysis, definitely, right?
It's an incredible living entity. The Gaia hypothesis is really strengthened by something like this, I think, which I adore that hypothesis.
Which is?
The Earth is a living entity that's self-regulating. All of the conditions on Earth that you see here are regulated by life forms. The amount of oxygen in the atmosphere, the temperature of the Earth, the recycling of nutrients, all these things. The geochemical.
It posits that the Earth is a meta-organism.
Yeah, but I mean, it makes sense when you look at all the activities of all the life forms. For instance, the geologists used to call these geochemical cycles. And in fact, Darwin started his life as a geologist, interested in how the volcanoes got to be islands and all that stuff, right? Mm-hmm. And later, when we discovered that there isn't a single process on Earth that isn't life-driven, all of those geochemists changed their terminology to biogeochemical cycles. Every single cycle is now a biogeochemical. And so now you have to take into account life forms in order to get cycling. And that's where we're really screwed up in our thinking, by ignoring that fact and saying the Earth will take care of itself. It doesn't matter what we do to it, it'll take care of itself. If you destroy things like coccolithic phytoplankton, you're destroying the ability of the Earth to make clouds, for instance. And so when you start to think about the philosophy of how life evolved to sort of take care of itself, given a substrate, here's your banked material. All right. You can draw on those resources all you want, but you can't use it up. You have to live within your means. You have to integrate to recycle stuff. Imagine how complex that looked no matter when you started. And here we are today trying to piece it all together to try to prevent us from destroying all of that. Right? And that's where we find ourselves. That's why everybody's throwing up their hands. And how can you deny the fact that the Earth is changing at a more rapid rate than it was before we were here? How can you
deny it?
How can you? But you do anyway, because it's politically correct.
It's not politics, it's fact. I don't have a pick related to that.
Oh, I hope so. So that's as a non-virologist that just oversaw this whole thing, I was very attracted to this one.
I want to just bring this back to the Klosnu viruses. One more thing here. So the original paper on Closnu virus, as we mentioned, was just a sequence. Now we have a related virus of a closely related to a Closnu virus, say. The reason why Closnu sequence was originally interesting is because they had more genes encoding components of the protein synthesis machinery than ever found before. And the phylogenetic analysis suggested that they were acquired evolutionarily recent as an adaptation to their host. So the idea that there was a fourth domain of life that gave rise to these viruses was not consistent with that. And now here we have an infectious virus related to that, which has diverse translation components, which were more recently acquired as well. And in fact, what's neat here is that this virus, in fact, has lost a lot of these, all of the tRNA genes, and because they're not needed in this host. So they say BSV provides evidence that the translation machinery is a homoplastic trait, need not be ancient in origin, and is not evidence of a fourth domain of life. Now, all that may be true, but I would have liked them to discuss it a little more, to flesh it out, right? Because all they're doing is bringing up the conclusions, and a lot of people will read and go, why? Why is this the case? But
right, so they're just saying Dickson is wrong.
A lot of people have said that, Alan. But it doesn't stop me.
They need to elaborate.
They should elaborate, yeah.
And I then also had to look up homoplastic, and I found that one way of saying it is that homoplasy is a product of convergent evolution, whereas homology is a product of divergent evolution. And so that was an interesting way to think about it, that homology and homoplasy are different in that regard.
All right, I think that's enough of that. Anything else anyone want to say about this paper?
Curiouser and curiouser.
Curiouser and curiouser. All right, authors, if you're listening, you can call Kathy and she'll say straight to
tell you what figures I still
However,
find confusing.
You have to give her an authorship.
No, you don't. Not even an acknowledgement.
An acknowledgement would be good.
No.
We have some email. First is from Niraj, who writes, "Hi, TWiVanguardians. It's been a while since I've written to TWiV, but as always, it was great listening to all the in-depth discussion on various issues concerning life as a scientist, especially in response to Raihan's email and pressures, frustrations of publishing. Amy also added to this a bit. Personally, having gone through the grind of a graduate school and postdoc, I can totally understand this predicament and frustration. Circumstantially, I have unfortunately worked in labs where publishing only in a CNS, Cells, Nature, or Science journal was the holy grail and pretty much the sole criterion for measuring your intellect. However, personally, I was always befuddled by this rationale that for science to be good and impactful, I need to make sure it has a CNS publication tag too. By the way, the tag in biology also manifests a stop codon. Geeky? Sorry, I digress, but in any case, this was a common discussion point amongst a lot of my fellow colleagues, especially during my postdoc days, but like me, they were also burdened with the same responsibility and outcome, i.e. to keep slogging for the elusive manuscripts in one of these damn top-tier journals. And to this day, this is something that continues to puzzle me, since how can where and not what be more intriguing in science? It's a great statement. In my humble opinion, highly reputable journals like JBC, PNAS, JCB, etc. do publish amazing scientific stories, and CNS journals at times do publish some really crappy ones. There, I said it. Having suffered through this personally, I have at times had difficulty even reproducing Figure 1A. Forget about the whole premise of the publication from the CNS journals, Nature in particular, for some reason. For example, on one occasion, we even made a knockout mouse based on the results of a publication. My postdoc lab at Genentech was apparently very well funded, just to find out that the observed genotype to phenotype effect was totally false, as the involved gene had nothing to do with it. That made the bleeding, so to say, excessive. It made me wonder as to why does one need to be elitist, so to say, when it comes to publishing high-quality work, and to make matters worse, why are scientists measured on those metrics? Clearly the system isn't working. No one really regulates or validates what gets reported in a journal anyway, so to make life and death situation based on where the work was published seems a bit foolish. Isn't the true scientific merit based on the impactful nature of the discovery rather than the impact factor of the journal where it is submitted for publication. And unfortunately, to make matters worse, a lot of scientists, either under the pressure of gaining tenure or addicted to glory points, are also to be blamed for the sustenance of this culture. If the reported science is fundamentally solid and can be reproduced for the most part, then that is a fantastic outcome as far as I'm concerned. And to that point, Rich, I'm glad you had a clean mindset of publishing relevance rather than rushing after namesake glory. Overall, I do realize that the difficulty in getting funding for exploratory science is probably getting harder, and it helps to get support from PIs who are giants in the field, but the amount of stuff that gets reported in CNS articles at times seems obscene. Why should one have to answer all the relevant questions? Isn't the fact that the reported work has generated more curiosity equally important? And to make things worse, this professional line of work doesn't at all pay you well. One probably spends close to 8 to 10 years of their prime being a grad student and postdoc. I'm talking more about biomedical sciences. And even at the end of it, there generally isn't a very well-laid path forward. And especially if you wish to venture into the biotech industry, it becomes even harder coming from academia. A lot of times the jobs demand some on-the-job experience, and without having been exposed to that facet of a research enterprise, how can one do justice? Due to all these reasons and an absolute lack of mentoring, unfortunately that has been my experience up until I started working in a startup, I feel science can be a very lonely endeavor. Everything seems positive when experiments work, but in a profession where 90% or more of the efforts fail, although one mostly learns from those, I think this whole charade of glory chasing through publication in high-impact journals seems depressing, counterproductive, and somewhat outrageous. But it's not all gloom, and there certainly is the upside of being engaged in doing something that is both invigorating and rewarding in a very tangible way. Overall, based on my experience, all I will say is that being in research will surely test your patience and resolve, but if you have the passion and the fight in your heart, you will overcome the limitations. For me, a life outside research would be a life without curiosity and fun of discovering the unknown. Personally, exploring the unknown is the biggest driver for me, and I hope when I'm done, I would have made some intriguing and hopefully interesting discoveries that can invigorate an idea and maybe an individual. As always, thanks to all you folks for being so generous with your time and effort. It is always special to listen to these podcasts and learn. Best Niraj, who has written multiple times and spent some years at Rockefeller as well, Dickson.
With whom?
Forgot.
And Niraj is now at Sutrovax.
Sutrovax.
One comment I wanted to make was there was a stretch of time when Cell was almost more known for the things in it that weren't correct. And so some virologists said the study section related to me how the saying in their lab was, just because it's in Cell doesn't mean it's wrong.
That's great.
Right.
That's great. And that changed for sure. Well, those are all great points for sure.
They truly are.
Echoed many times. So thanks for your thoughts. Alan, can you take the next two?
Yes. Bill just sends a link. He says, finally killed for good. And this is an Ars Technica article about the Electronic Frontiers Foundation legal battle with somebody who claimed to have a patent on podcasting.
And we would have had to pay them.
Yeah, it was basically a patent troll story.
We are grateful to the EFF for doing this. Yes. So now we can podcast Dickson.
We can podcast Dickson?
Yes.
Yes, and we are podcasting Dickson right now.
My goodness.
But more importantly, this ridiculous patent claim does not get to charge people for podcasting.
Yeah, we would have to pay royalties to them.
Yeah, which is absurd because they didn't create anything.
Correct.
All right. Next up, Theodora writes, "Dear all, I hope you're well, and the weather everywhere is good." I think she wrote this a couple of weeks ago. I'm following up on the discussion over capping the number of NIH grants per investigator from episode 467 that Alan and I have continued on Twitter. Let's start with and by the way, Theodora and I were going back and forth on Twitter. You can follow the thread of that there if you go to my Twitter or hers. So she and I disagreed was the upshot, and we discussed it there. And I said, you ought to put this in a note to TWiV. Okay, so she says, let's start with the fact that ultimately funding of investigator-initiated proposals is decided by their peers, and that these ever-changing committees of scientists known as study sections are not composed primarily of old established investigators. In fact, the study sections I've been on have been the complete opposite, and in my experience were not at all biased when evaluating applications from big labs. The NIAID pay lines also favor young investigators. Their applications have to be in the top 13% to obtain funding, whereas everyone else, this percentile drops to 9%. Importantly, the reports on which Alan has based his opinion that big labs do not produce the most impactful papers have several flaws. One such major flaw is that they only consider the NIH or equivalent funding of each lab in their analysis. Therefore, labs that have other sources of funding, Howard Hughes Medical Institute being the most prominent, are misplaced on the graph and thus skew the results. Furthermore, in the PLOS One study, the data are plotted on a log scale, which diminishes differences. What I've not heard being mentioned in this debate is a recent shift in the NIH funding from investigator-initiated proposals to multi-million-dollar pet projects selected by administrators. I think this is a problem as it reduces funding that allows investigators, young and established, to pursue what they think is important. And on that point, I think Theodora and I are in complete agreement. Ultimately, increasing this latter pool of money is key, and I wonder why we turn on successful big labs whose funding is actually a tiny fraction of the total NIH budget, rather than consider the NIH budget itself and work on ways to increase the part devoted to investigator-initiated research. There's no point funding young investigators without a plan to continue their funding once they are not young anymore. There has to be competition, and track records matter. This is not a perfect system, but it is one most based on merit I have experienced. It is what allowed for my own success while not being rich, evil, privileged white male head of a big lab. And I am in no way a cherry-picked example. Okay, points taken, Theodora. I think we disagree on the interpretation of the data, the NIH grant data. But I think we agree completely on the diversion of huge sums of money to big science projects when that might have been better spent on funding more R01s.
Well, I have anecdotal data, which has been over the years, every big lab I have seen, yeah, they produce good stuff, but there are an awful lot of people there that don't produce anything.
Right.
So is that the overhead for producing something that you have to have per every paper? You have to have 5 people that don't do anything? I don't think so. Right.
No.
I do agree that the big pet grants are a big problem, and shifting away from R1s, I don't really like that at all. I mean, part of the issue here is the attempt to spread out the money, and there was an attempt to cap the number of NIH grants, which a lot of people didn't like, and that got thrown by the wayside. And so the idea is, let's just better review what we're funding, and that means better study sections. And I think the study sections are terribly flawed. Okay, overall they do some good, but there are always issues at every level. I've been on them for many years, and you know, it's great to have a range of expertises, but you have people that don't really know enough to be able to judge, or they're too critical, and I think there are big problems with the way it's done there. So I'm not sure that's the solution to this. I don't know what is. I just don't think anyone should have 11 R01s. If you cap it at 4, then that's 7 that other people get to have. But right. I understand that a cap is not wanted by anyone, so be it. I don't know what the solution is. And it's
particularly not wanted by the most powerful people in the field, which was my point.
The thing that always frosted me over was you have to list the percent of effort that you put into each grant. And if you have more than 3 grants and you say 50% effort for each grant, that's a little bit like a Yogi Berraism.
Yes.
And I'm really offended by that, because everyone knows that they're not going to do it that way, but they give it in the grants anyway. So,
well, if you had 11, you'd have to have less than 10% effort. And I think that's
how could you get a grant with 10%?
Well, that's farcical to say I only need to put 10% of my time to make this. It's crazy.
Well, you know that some other person is in charge of the actual running of the grant then, right? So it's not fair.
What is the actual amount of money that is consumed by people with more than 5 R01s per lab? Do we know this?
It's a lot.
Some people have told me it's not much.
You think?
Like Theodora, who is Theodora Hatzianu.
Yeah, she's saying it's not much, but I don't know what the numbers are, and I'm not sure that she knows what the numbers are either.
Because it's a small percentage of...
But what is that percentage?
I don't know. I do agree that the pet projects are really a problem.
Yeah.
There you have big labs who are really not productive running off these.
Are you talking about program projects?
Yeah, program projects, excellence, centers for excellence and so forth, where essentially administrators decide that this is where the money should be going. And that bypasses peer review, although they are peer reviewed.
No, they are peer reviewed.
Yes, but they're pet projects selected by administrators.
Yeah.
But I agree, they're peer reviewed. And so if they're no good, they're not going to
They're so complicated to put together that only a few labs will be able to do that, actually. It's not a highly competitive grant situation in that sense. Only the big groups can afford to get involved.
Yeah.
And they designate people to write them up, and I've seen them. I've been on several of those things. I've rarely seen them get turned down, by the way.
There was also a suggestion, I think it was Arturo Casadevall, came out with this a couple of years ago, and nobody took it seriously, but I think it was intended seriously, and it was actually kind of a good idea. There's pretty good evidence that study sections are very good at identifying about the top third of grants.
Correct.
Yeah, that's right.
The problem is dividing it into finding the top 9%, the top 10%. Because when you go from the top third down to the top 10%, now you're really splitting hairs and saying, this lab is more likely to do this experiment seems a little more interesting, and you're really making qualitative judgments. And so the suggestion was, well, why not just assign the study sections to designate the top third of grants? And then once you've done that, you have a lottery.
Right, and
from that
have
top
it be
third.
a total random lottery,
right?
As opposed to the situation now where it's inconsistent. From one time to the next.
Right.
If you resubmit a grant, you're not going to get the same reviewers, and so it's a lottery as well, but it's not a randomized lottery.
It's not a randomized or fair lottery. So if you had it, if you said, okay, here's the top third, now we're going to take 10% of this, group, and we're going to give them grants, and so we'll pull the names out of the hat, and here they are.
So it would be a crapshoot.
It would literally be a crapshoot. Well, actually, worse odds probably, but you'd have roulette odds on getting your grant once you got into that top set, and that would actually be a fairer system. But nobody took it seriously, and so I don't know.
No, because they didn't want to be accused of having a crapshoot to determine the top grants.
I think the reason it was disregarded so quickly was that nobody wanted to admit that it really has come to that. But another advantage of a system like that is that if donors come along, the Gates Foundation or the Zuckerberg Foundation comes along and says, we'd really like to support some good science. Hey, guess what? We've got these projects that have already been identified as good that we couldn't afford to fund, so pick some.
Or pick some more out of the hat.
Pick some more out of the hat. Sure.
We talked about this with Arturo Casadevall on TWiV 154, if you want to hear him talking about it. It was him. That's what I want to mention one thing. I was recently at a meeting concerning one of these types of center, and a official made the statement that next year, this particular type of center, a big multi-investigator center, what Theodora calls a multi-million-dollar pet project selected by administrators, they're going to be cut 20% next year. And we said, why? And they said, because the R01 pool is shrinking and we can't have that happen. So that's a good sign that there is some balance kept in mind. They want to maintain a certain pool of R01s, and they're going to cut these big programs to do that. I like that.
Or they could sell one atomic nuclear submarine.
Yeah.
They could sell one?
Just one.
How much is that?
It's enough to fund NIH for a couple of
billions
decades.
And billions of dollars.
They want to increase the defense budget.
I know. They should do that with R01s.
Well, I think a creative way would be to have the Department of Defense fund some basic science research, right? Which they do, as long as it's involved with their goals.
Yeah, it has to be
They actually have a pretty broad interpretation of that. They do fund a lot of basic research, but it's...
I mean, that's where most of the breast cancer work comes from, right?
Is that right?
Yeah, the DOE. Mm-hmm. That's right.
Mm-hmm.
Department of Energy?
It used to be DOD.
Mm-hmm. Okay. Kathy, can you take the next one?
Anthony writes, And he just sends a link to Rabies Alliance World Rabies Day, which was September 28th.
Long time ago.
He writes, sad to relate, I only learned of this yesterday. Well, sad to relate, we only read it today. The website does show some events for this week. If this was mentioned in TWiV 461, I'm sorry for an unnecessary email. I'm just up to 87 minutes. If it was not mentioned, was the choice of paper covered serendipitous? Yes, I know that the text readability checker and Gerard Manley Hopkins would say that 5 syllables is way too many, for what it's worth.
461 was using rabies virus to trace neural circuits.
Right.
Well, of course, we picked it exactly for that reason.
Absolutely.
Because it was World Rabies Day.
Absolutely.
Yeah, thank you, Anthony. Yep. Okay, you can take the next one also.
Or writes, my name is Or, and I am a BA student in biotechnology from the Technion in Israel, and I'm on my way to becoming a virologist. And I want to tell you that virologist is capitalized. So it's important. First of all, I wanted to say that I'm a fan of TWiV and of Professor Vincent Racaniello's virology lectures on YouTube. I've watched them prior to the virology course I'm taking now, and I found them to be a very thorough guide into the world of viruses, as they greatly prepared me. I want to ask of you if you could do an episode about examples on the positive effect viruses have on nature. In a recent lecture during my virology course, we discussed a few examples of traits that viruses grant their hosts, like the well-known case of cholera and its toxin. All examples were negative, so when asked if anyone knew of anything else, I pointed out a positive example from Professor Racaniello's lecture about Yellowstone's plant Dicanthelium languginosum and its virus-infected fungus. I would like to have a better arsenal of examples, even if there are only a handful, for the positive effect viruses have on nature, besides, of course, evolution. I found an ASM entry from 2015 that said, mammalian viruses can also provide immunity against bacterial pathogens. Gamma herpes viruses boost mouse resistance to Listeria monocytogenes, an important human gastrointestinal pathogen, and to Yersinia pestis, otherwise known as plague. Humans are often infected with their own gamma herpes viruses, and it is conceivable that these could provide similar benefits. And he gives another link to that ASM entry. However, my search yielded nothing more that was of relevance to my query. Thus, I would like to ask you, could you make an episode on the subject? It would be very educational for many people. Thank you for taking the time to read my email, and I look forward to future episodes of TWiV. Sincerely, Or. And I remembered TWiV 92, the one that was live from Bozeman, Montana at the ASV meeting with Marilyn Rusink, who discussed how plant viruses make plants resistant to adverse conditions. And it's a pretty short episode, 57 minutes. So you could check that out. But obviously, TWiV 92 was 2010, so that's a while back. And then someone else has included that Ken Cadwell and the effect of mirroring noroviruses on development of intestine and the immune system.
Yeah, I think we did that on that paper on TWiV also. That's a pretty cool example. You know, there aren't huge numbers of examples. It's a hard experiment to do.
They don't always make you sick.
I mean, all these genes that we have from retroviruses, many of them we know help us, but does that count as a beneficial virus? Sure, why not?
Sure.
I was talking with someone at ASV this past summer about this, and his thing was, well, there aren't that many examples because no one's found them yet, and that may be, but certainly Maryland has lots.
We've looked at the ones that affect us, make us ill.
Yeah.
But there was the one with the parasitoid wasp.
It's good for the wasp, right?
Yeah, it's good for the wasp. It's bad for the caterpillar, right?
Yeah,
that's right.
Yeah, there are a bunch of those kinds of examples out in the insect world for sure. Yeah. Do you think we could do a whole episode on it? Just go through. I don't
know.
Maybe we'll see, or maybe not.
We don't know.
Dickson.
Josh writes, "Good eye, TWiV team," which is the way they would say that.
How you say it?
Good eye. Good eye. They don't say good day.
I'm going to get letters.
They say good eye.
Okay.
My question relates to TWiV 457. I really enjoy learning the origin of words. It helps me remember the disease pathogenesis and brings meaning to the scramble of Latin binomial. What is the etymology of myxomatosis? Growing up in rural Australia, I always thought that the scientific name for myxo was myxomesothelioma. This is just some misspoken version of the actual disease name, or does it have an actual base in science? Hopefully you can reassure me, since I've been telling people for years that it's myxomesothelioma. Whoops. Thanks for the show. I work a very mundane lab job, and listening to TWiV helps keep my mind moving while I work. Josh.
We've had some emails, and especially from Rich, and
I think they came after TWiV 457. So Josh keeps listening, or has kept listening. We had that as an arc on 2 or 3 episodes about.
Yeah. I think the mesothelioma was that the lesions look a little bit like mesotheliomas, and that so that may rabbits. Who's the virologist at Arizona now, the pox virologist?
Grant McFadden.
Grant McFadden wrote and explained that origin. It's in one of the TWiVs after 457.
That's right. Josh. Keep listening, Josh. You'll get it eventually.
Yeah.
All right.
Good on you.
Let's do a couple more. Anne writes, "Hello, TWiV team."
And by the way, mesothelioma would be very much a topic in rural Australia too, wouldn't it?
Yes, indeed.
Because there were asbestos mines.
Asbestos mines.
I remember something about that. Anyway.
Could be.
Hello, TWiV team. I'm months behind in listening to the TWiV, but the dispiriting political news of late has encouraged me to focus on constructive conversations that can help keep my focus on curiosity and learning. This was an excellent episode, especially the discussion of plaque assays. I still haven't taken Dr. Racaniello's virology course, hopefully remedied in the new year, so I was pleased to hear the discussion of the techniques and Dr. Despommier's questions, some of which were the same ones I had. As a former student and science teacher, I appreciate the questions and the team's willingness to answer them. Thanks again for all the work you do to bring science to the rest of us. It is much appreciated. P.S. I went looking for some of the movies showing viral action in a plaque assay, and unfortunately most of the links don't work. And she is linking to my virology blog post. Now playing viral plaque formation, at least in Safari on my iPad. I did find the more recent Virus Watch episode on YouTube where Dr. Racaniello described plaque assays, and which included one of the films. I will remedy any link issues, Anne.
I went in and clicked on them from my desktop computer on Safari, and they did work there.
Oh, yeah? So, did they actually play?
Mm-hmm.
Okay.
Maybe the server matters then.
Alan?
I'm going there now in Firefox and trying to check. And I'm downloading something that wants me to play with QuickTime Player. I think that's the issue on an iPad.
QuickTime Player, that's right.
It wants to download the movie to my desktop. And it's coming in as a QuickTime movie that downloads. And I think on an iPad, that's not going to happen.
Yeah, I clicking on it here in Safari on a Mac, and it just downloads it. So
yeah,
it plays. Yeah.
So I should set it up so it plays. We will do that. It's easy enough. Okay, Alan, can you take the next one?
Yes. Just get to that. Bob writes, story spotted while browsing. Quoting the story, the U.S. Environmental Protection Agency has approved the use of a common bacterium to kill wild mosquitoes that transmit viruses such as dengue, yellow fever, and Zika, Nature's News team has learned. On November 3rd, the agency told biotechnology startup Mosquito Mate that it could release the bacterium Wolbachia pipientis into the environment as a tool against the Asian tiger mosquito, Aedes albopictus. Lab-reared mosquitoes will deliver the bacterium to wild mosquito populations. The decision, which the EPA has not formally announced, allows the company, which is based in Lexington, Kentucky, to release the bacteria infected in mosquitoes in 20 U.S. states and Washington, D.C. Mosquito Mate will rear the Wolbachia-infected A. albopictus mosquitoes in its laboratories and then sort males from females. Then the laboratory males, which don't bite, will be released at treatment sites. When these males mate with wild females, which do not carry the same strain of Wolbachia, the resulting fertilized eggs don't hatch because the paternal chromosomes do not form properly. The company says that over time, as more of the Wolbachia-infected males are released and breed with the wild partners, the pest population of A. albopictus mosquitoes dwindles. Other insects, including other species of mosquitoes, are not harmed by the practice, says Stephen Dobson, an entomologist at the University of Kentucky in Lexington and founder of Mosquito Mate, and gives a link to the article.
What do you think of that, Dickson?
It has promise. Yeah, but here's a confounding factor here. That's A. albopictus is far less susceptible to dengue virus than Aedes aegypti. A. albopictus outcompetes Aedes aegypti for breeding sites in transition areas between the south and north.
Take albopictus away.
So if you take albopictus away and the other mosquito may come back.
It's not even a good host for Zika, right?
No.
To kill wild mosquitoes that transmit Zika, nah.
I think albopictus is the wrong target. I think the right target would be Aedes aegypti.
Anyway, continue.
Sure. Bob says, P.S. While watching TWiV 486, the Capsid Club, or 466, the Capsid Club, I was intrigued, as I have been in the past, with Vincent's comment to the audience, something like, how many of you work with double-stranded RNA viruses? I guess that someone choosing to work with a given virus or a family of viruses had to choose it on the basis of practical considerations. RNA viruses mutate more frequently. Is there a model host in which the virus will propagate, etc.? Funding considerations, popularity, how crowded the field is, etc., etc. Might this make an interesting future TWiV episode or sub-episode? Yeah.
Yeah, it's a good idea.
Yeah, why did any of us choose?
You should do a little sorting hat quiz on your website to sort people out. Which viral house are you in?
Yeah.
Right. Who are your people?
But one of the things that Bob doesn't mention is, what has the best way to study the questions that you personally find interesting, yes, which is one reason why you might end up with a particular virus. And another way is that when you're mentored as a postdoc or as a graduate student, the virus that your mentor works on might be of interest, but it's more the mentoring. And so you choose that lab and then you know that virus. So then you keep working on that virus or related viruses, absolutely, or you get as far away from that kind of virus as you can. So there's, some more scientific interest in questions, I think, that relate to it.
Mm-hmm.
Yeah, Peter Palese said to me, you want to work on RNA viruses? I said, yeah, for a postdoc. He said, then you have to go to the best lab.
Mm-hmm.
And I said, who would that be? And he said, Dave Baltimore. So and I was okay with polio, but, it's not like I picked polio, but RNA viruses.
Yeah.
All right, let's do two more.
Is Kathy next?
Wait a minute. Which one did we just... Mosquito Mate, that was Alan. That's Kathy's next. Yes.
Amir writes, Dear Vincent and other TWiV hosts, thank you for your fantastic podcasts. I recently ran across a short story by Ted Chiang, who is a multiple award-winning American science fiction writer. The story is very short. It doesn't take that much time to read it, and it has been included in the Best American Short Stories 2016. And he gives a link to the story, which is entitled The Great Silence. And I read the story. Doesn't take very long to read, and you might want to check it out.
I read it to Dickson before, right?
You did.
It's about a parrot, right? No, a gray parrot.
Mm-hmm. Right.
Mm-hmm.
Right. I guessed it was a gray parrot. You just said parrot. I said, was it a gray parrot?
Why did you guess it was a gray parrot?
Because I've
smart.
They're very
smart,
and they live a long time.
Maybe I should get some.
Maybe they make great postdocs.
They'll outlive you.
Really?
That's true.
They're going to live like...
They're low-maintenance postdocs.
20, 30 years.
They live decades.
They do.
They'll certainly outlive Dickson.
They'll definitely outlive.
That's not nice.
No, it really is.
I hope you're around forever.
Oh, I don't hope
Don't you become a vampire? I hear you could join the vampire club. All right, one more, Dickson.
John writes, I am a physician and a writer who is interested in writing a science fiction thriller about bioterrorism. One thing that worries me is that advances in synthetic biology may permit the reconstruction of a virus such as smallpox, which can then be weaponized by simply introducing a suicidal vector like a suicide bomber into a population. I note that the genome of the smallpox virus is in the public domain, and while synthesis of a full genome may not be immediately possible, one can imagine that some sort of piecemeal approach may be possible presently. Horsepox was recently reconstructed. One can readily imagine that from scratch synthesis could be possible within decades. Vincent, you had a reputation for that, didn't you? I do not want to give anyone any ideas, but it seems to me that the best defense against such a threat would be a public appreciation. Can you comment on what precautions are in place to prevent this from happening? Please let me know if you plan on addressing this on your show. Thanks, John. Well,
see the movie 12 Monkeys.
What's that?
Oh, that was a good science fiction movie. Was it Bruce Willis?
Yes.
gets sent. He's a prisoner in the future, and he gets sent back as a time traveler to various times in the 20th century to find out the source of a mysterious outbreak that has decimated humanity, and this, he's getting sent back from this decimated world to find where this epidemic came from, and there is very much a story like this behind it all.
Well, the synthesis is now, it's here. It's not within decades, because this horse box it's big.
Within weeks, yeah.
And so we have regulations in place to prevent scientists from doing this, right? We have, if you wanted to synthesize smallpox,
an evil scientist.
You couldn't. Of course, someone who is not within the bounds of regulation could possibly do it.
A disgruntled scientist that didn't get their grant, or
someone else who wants to use it.
Correct.
So that's a problem. But I don't think, you say it can simply be introduced into a suicide vector. It may not be so simple, right? You take an infected person. So let's say a volunteer says, I will be infected with smallpox and walk into New York City. I don't know how effective that would be at spreading it, right?
The problem with a lot of these viruses and bacterial infections too is that they show symptoms very soon after you acquire them. So there's no delay, and then therefore you can identify the outbreak, and therefore you can start quarantining. Like AIDS, that's what took AIDS. That's why it had an advantage, right? It took like 8 years or something like that to manifest itself in terms of clinical symptoms. So you need something like 3 to 5 weeks after exposure, you start to suffer. And that's, I don't know anything like that.
Well, this is why we stockpile smallpox vaccines and antivirals, okay? Because smallpox is a good, is a good bioweapon virus, because we don't immunize in the general population any longer. It's quite lethal, 30% lethality, right?
You need more than that for a bioterrorist.
But we can't do it. My point is, scientists cannot work with it. So if anyone's going to do this, it would be outside the realm of regulation.
And when you're outside the realm of regulation, you're going to have to get access to this very tightly guarded virus. And the phrase you use in the letter, John, where you say reconstruction of a virus such as smallpox, smallpox is in its own category because it has been eradicated from the wild, and we haven't done that with any other human virus. So it puts it into a different situation from anything else. This is not like polio that we're still actively immunizing against. This is not like any other disease that we deal with routinely. And as a result of that, a huge portion of the vast biodefense industrial complex has been configured around dealing with smallpox, and tens of millions of doses of smallpox vaccine, the TWiVax formulation, are stockpiled in multiple locations around the United States, ready to be pushed out and air freighted. And this particular scenario is one for which the system is extremely well prepared. Other diseases, maybe not so much.
Like the 1918 influenza virus.
Yeah, 1918 flu coming back, we're a lot less prepared for. And that is actually something I would be a whole lot more worried about.
Yeah, that's right. I agree.
But it's also something that's would happen naturally rather than being deliberately done by humans.
Yeah.
I think we have talked about this often on previous TWiVs, John. So, I know I can understand you might not want to go back and listen through all of them. But we have, maybe you can find no, you won't tell from the titles. That won't help.
Not necessarily. No,
you'll have to look at the
Sorry
show
about
notes.
that.
But smallpox is good for this because it effectively transmits person to person, fomites, respiratory transmission, whereas Ebola wouldn't be as good because you need pretty close contact with secretions, right? So not every virus would be well suited for this. But, John says, I don't want to give anyone ideas, but it seems to me that the best defense would be a public appreciation. I think the people who want to do this already have the idea, right?
Yeah.
So there's nothing new, and us talking about it isn't going to help.
And by the way, from a plotting perspective, John, I don't know who the characters are who carry out this bioterrorism in your science fiction thriller, but you probably ought to make it some kind of a doomsday cult that doesn't care about consequences. because any terrorist group that's trying to impress themselves as being the champions of the poor or the downtrodden or whatever would immediately appreciate that releasing something like smallpox in the world would really hurt the poor and the downtrodden a whole lot more than it would hurt, say, developed countries. Because we're the ones who can stockpile the vaccine, and the people who would really get screwed, since this would spread globally very quickly, would be the folks in the poor countries who wouldn't get the vaccine.
John, in your research for your book, you might look into the 1947 outbreak of smallpox in New York City.
Yes.
Apparently, a single gentleman was infected. This is a time when smallpox was still around. Was infected elsewhere, came into New York City. I believe he developed smallpox and was diagnosed, and then they massively immunized much of New York City's population. And I don't think there were many, if any, cases, but it would mimic a situation where you would send someone.
There was one additional. In fact, that was the case. I think that was the basis for saying smallpox is highly contagious, has high airborne transmissibility. There was one additional case of somebody who was like on 3 floors away from the guy in the hospital.
Yeah, I just found my blog post of 2009. There's a New York Times article there. Smallpox in city inoculation urged 3 cases reported.
3 cases
from this man who came from Mexico City with the illness. He died 2 days after being hospitalized, and that was it, right? 11 days later, a 22-month-old baby and a 25-year-old man were admitted to the same hospital with symptoms of smallpox, and they were positive for the virus.
Right.
So we responded pretty well to that. 5 to 6 million people were immunized by the end of April, and ring vaccination basically stopped the outbreak. So there is an article in Emerging Infectious Diseases. I will put a link to the blog post, John, so you could check that out. You might want to.
But I would say go with flu, not smallpox.
Yeah, me too.
Which flu strain would you like to use?
Oh, pick one. Make one up. It's fiction.
Yes.
Oh, yeah, but you'd have to be a lethal strain, not a comparatively mild.
The one that killed all the mastodons.
Yeah, right.
Right, there you go.
All right, let's do some picks. Alan, what do you have for us this week?
I have, this is about as nerdy as it gets. Well, I shouldn't say that. I may top myself at some point, but this is the shortwave radiogram. This was, Voice of America was doing this for a while, and then I guess they ran out of funding for it, and now the guy who had been running it for them, Kim Andrew Elliott, decided to just go on his own, and I think he's crowdfunding the airtime at a couple of shortwave radio stations, WRMI in Florida, and then he's got a couple of other stations that broadcast it as well. So shortwave radio is the area between your AM dial and your FM dial, and there are several shortwave bands you can tune to. This thing broadcasts on 9400 kilohertz and 7730 and 11580 kilohertz. Shortwave radios are easily available. You can buy them on eBay or even at electronic stores for $100 or less. And you can tune to this thing. What's cool about it is, unlike everything else on shortwave, where it's somebody talking, into a microphone or in some cases music, this is a broadcast that is sent digitally. And so you can use a phone app or run an app on your computer that will process the audio and turn it into text and images.
Cool. That's amazing.
And he broadcasts different formats to see what the best formats are, best digital formats are for sending this stuff. And on the website, he describes various reception reports that people have gotten, and here's how well it was received in Germany. Here's how somebody in Brazil got it, and that kind of thing. Because the great thing about shortwave is one transmitter in one location can reach the entire world without requiring any infrastructure in between.
Cool.
That's pretty neat.
Yeah.
Yeah, it is pretty nerdy.
Yes. Yes.
But you're right. You will outdo yourself, I'm sure.
At some point I will.
Something else. Really cool. Dickson Despommier.
Yes, I've got some more visuals, and I've gone back to my Cosmos magazine resource for this, but they're really great. I mean, you can't go wrong on these things anyway. So this is a potpourri of from the microscopic to the galactic. And the first one looks like a flower garden, basically. And it's actually not a flower garden at all. It's a microscopic image of fibroblast cells showing the induction of cell fusion by a newly described gene and its protein called myomerger, which I never heard of before, but it's interesting to see it at work. And I was, I'm always taken by these things anyway. So you got the sun getting loopy. You've got
that's cool.
The heart of a zebrafish, and then you got the face of an ancient native, and of course, and the new vision of the brain.
That's pretty cool.
And that's
that's an artistic depiction, right?
Yes.
I like the zebrafish heart. I think that's gorgeous.
Very neat. I mean,
I like the glass frog.
Frog,
me too, cool. me too.
Nothing to hide. Nothing to hide.
Me too. And the pollen, follow the pollen. Well, whatever. And then finally, something for Amy. I thought of this little tiny brainlet. Mm-hmm. It would be a good model for her to look at Zika with, I thought.
Don't tell her that.
I'm not going to tell her that. No, I won't tell her that. But it's a human brain in this case.
It's an organoid.
It is, but it's human.
Yeah, it's okay. You can human stem cells. You can make them. The reason I say this is because when we get our grants reviewed, they always say you should use organoids, not slice culture. So
why not both?
Well, I think that may be the key, Dickson. Sure, we'll do whatever.
This photo of Jupiter looks like Van Gogh's Starry Night.
Yeah.
It's gorgeous.
It's got so much detail.
Mm-hmm.
I mean, it's great.
Are those gases? Those swirly?
They are.
Swirlies.
They are. Versions of hydrogen and CO2 and some methane thrown in there.
This is from the recent mission.
Juno. It's from Juno,
right?
Yeah, that's right.
It's cool.
It's quite remarkable.
Cosmos.
And by the way, that Juno mission, just as an aside, discovered a new radiation belt on Jupiter, which is at the equator.
Yeah.
So
cool.
Lots of interesting stuff out there.
Kathy, what do you have?
I picked a site that's called Every Second. And earlier in the show, we were talking about something that happened to every second, and I swore I was going to remember it and bring it back around to this, and now I've forgotten
killing of cells in the ocean.
Oh, this is cool.
Yeah. But so when you first go to the site, there's a bunch of different tabs that you can push for asteroid forecasts, or the evolution of brands is an interesting one. Animal heartbeats shows you beats per minute, starting from the blue whale that only has 6 beats per minute all the way to the hamster beating at 450 beats per minute. So it's just a lot of fun things.
Why don't they have TWiV downloads?
Well, they could have that. Yeah.
They should allow you to customize it.
Yeah. And it says you can follow him to be notified about new visualizations, and I suppose you could tweet him and ask him to tell you downloads of TWiV or something like that.
It wouldn't go very fast per second.
Right.
Don't click the McDonald's one if you just recently had one.
But
a lot of I clicked the Apple one. That's pretty cool.
That is impressive.
And the evolution of brands is interesting too. It starts out with just a few brands, and then as time goes on, adds more brands, but also the logos of the existing brands change while you're watching.
Yeah. We need the number of infections per second in the ocean.
Yes.
That would be good here. It would be the biggest number.
Or you could have it in different environments.
Yep.
In the ocean and forests. And
That's right.
Cool. That's very neat. All right, I have a very simple pick, and it is the Instagram account of Science March DC. And the reason I picked it is because today they had a wonderful picture of Neil deGrasse Tyson and a quote saying, "People who deny human-induced climate change are badly misinformed. This position is neither politically liberal nor conservative. It's factual." And I just love that. It's a great, simple message. And if you follow them, they will have, now and then something pithy to say. The link is instagram.com/ScienceMarchDC.
Yeah, in the row below that, the
we
one
were
by
there.
Harrison Ford is too.
Yes.
So
That's excellent.
check it out.
Yep. And we have some listener picks. We have Islam, who writes, "Dear TWiVers, I came across this pic through a Facebook post by ASV on Sunday, December 10th. From the first glance, it looked like a virology crossword puzzle. I paused for a second before opening the link and thought, wow, this is very cool. After taking a careful look, it turns out that these are crossword puzzles created by Dr. Kevin Maringer, a lecturer of microbiology at the University of Surrey. It seems he creates them based on the lecture material to quiz the knowledge of his students. What a great teaching tool. And he links to a sample.
It is.
I was curious to know how he generates them. Turns out there is an online tool called Armored Penguin, which will do it for you if you can come up with words and associated clues. And he links to the puzzle maker. I have a suggestion for TWiV. For the book contest episodes, how about instead of asking the interested participants to simply send an email, ask them to solve a puzzle based on the topics discussed in that episode. I know this will come with an additional burden of creating and grading the puzzles, but you are the professors, and this is what you do, right? I always felt that just sending an empty message to win a precious book by random chance is not fair. Perhaps the TWiV crew can run these puzzle contests once in a while, which should add a lot of fun to the process and draw the winners out of those genuinely interested TWiV fans. I can't thank you enough for all you do to enlighten us. Also, kudos to Dr. Kevin for turning virology quizzes into fun. All right, let's come back to that. I think it's a great idea. And Peter writes, greeting TWiV team. I have a listener pick of the week. An article from Gizmodo where reporter Jennings Brown interviews some of the last people still using iron lungs and talks about their experience and the difficulties they have maintaining their aging machines in working order. It is a great article I saw some time ago as well, with good pictures of people still in iron lungs. They do exist. They're out there. So check it out. I think the crossword is great. I happen to have a crossword, which I got a couple of years ago, a virology crossword puzzle that Ann Palmenberg had made. And I'm going to post it on Sunday in the show notes. So you're going to have to go to microbe.tv and go to the TWiV show notes. There'll be a PDF and you can solve it. And if you solve it and send it in and it's right, I'm going to give you a book. I'm holding it here. It is called Neurotropic Viral Infections, Volume 1, Neurotropic RNA Viruses. It's edited by Carol Reese, and it's brand new, not even cracked, and it has articles on polio, measles, henipaviruses, rabies, neurotropic coronaviruses, neurotropic alpha viruses by Diane Griffin, and many, many more. A great compendium. Can be yours if you solve the puzzle. What do you think?
Sounds great.
So I have this puzzle already made. That's why I'll use it. But in the future, we could try making ones. I don't know. I'm going to check out this puzzle maker and see how hard it is. I have to solve the puzzle myself first because I don't have the answers to it. Hopefully I can do that. That's TWiV 474. You can find it at microbe.tv/TWiV. Send your questions and comments to TWiV at microbe.tv. Please consider supporting us financially. Go to microbe.tv/contribute. Dickson Despommier. ParasitesWithoutBorders.com. Thank you, Dickson.
You're welcome, Vincent. Happy New Year.
Happy New Year. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks, everybody. This was a lot of fun today.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for supporting TWiV and Ronald Jenkins for the intro music, ronaldjenkins.com. Happy New Year to all of you,
indeed,
my co-hosts, and all of you listeners.
Yep.
Hope that 2018 brings you lots of beneficial viruses. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.